Hi, everyone, and welcome to Microbial Minutes. This is the American Society for Microbiology's every couple of week update on what's hot in the microbial sciences. I'm Julie Wolf, and this is our C. diff is a pain session. Today, we'll be talking about five different topics. The first is painkillers and C. diff infection. Next, we'll move on to some antibiotic resistance and how it correlates with antibiotic use. Then we'll talk about some improved anti Ebola antibodies, which may prove useful in the ongoing outbreak in the Democratic Republic of the Congo. Our fourth story will be a story about viruses and quorum sensing, which is pretty novel and cool. And then finally, we'll end with a little bit about DNA in space. Jumping right in, we're going to talk about a story from MBio with the take home message that common painkillers may exacerbate C. difficile infection. So, C. difficile is something you are likely familiar with. This is the most commonly diagnosed cause of antibiotic associated diarrhea. And it's antibiotic associated because C. diff can form these structures called spores, which I'm showing you here on the right hand side. And those are metabolically inactive and extremely structurally strong. And so they are able to resist the action of antibiotics, which often require that bacteria are dividing in order to kill them. And once the neighbors are eliminated by antibiotic, the bacterial neighbors that cannot form spores, this opens up a niche that C. difficile can grow into should the antibiotics go away. This allows C. difficile, which is often a normal component of many people's gastrointestinal microbiota, to overgrow and cause disease. C. difficile infection. And I want to point out here that this is no longer Clostridium difficile, that the genus name has changed. It's Clostridioides difficile, but we can still say C. diff or C. difficile. This type of infection is the most common healthcare associated infection in many United States hospitals. And this is in part because of the recurrence rate. So oftentimes a patient will be treated with these drugs. They will dampen down the symptoms and perhaps even eliminate the diarrhea, but they won't get rid of all of those different C. difficile spores. And this very tough to treat recurrence is one of the reasons why there are nearly 30,000 deaths annually within the United States from C. difficile infection or CDI. Now, another common thing that people use in hospitals are nonsteroidal anti-inflammatory drugs or NSAIDs. These are extremely common, both over the counter, as I'm showing you over here. Ibuprofen, Advil, we're all familiar with these. But there's also a number of prescriptions that are written annually, including the drug that they are testing in this study, which is indomethacin. These are used for a number of different purposes. They can be fever reducers, they can be pain relievers. Depends on which exact molecule you're working with, but they're generally known to be COX inhibitors, which are cyclooxygenase enzymes that do a number of different things in our body. But one of the things that COX enzymes do is to generate prostaglandins. Prostaglandins are a molecule that is important for a number of different things with gut health. And so using these NSAIDs for various different reasons can have negative effects on gut health. This was already known before this study was done. But what this research group wanted to ask was whether taking these NSAIDs for perhaps any number of reasons could have a negative effect on the outcome of a C. difficile infection. And so they tested this in a mouse model. And what they were able to find is that indomethacin treatment does lead to prostaglandin deregulation, as was already known, and this can change the transcriptional and protein profile of different pro-inflammatory cytokines and chemokines. So I'm showing you over here one of the figures from their paper. You can see in this heat map, antibiotics and antibiotics plus indomethacin have actually a really common expression pattern, whereas antibiotics plus C. difficile does change this expression pattern from that of antibiotics alone. But once we add indomethacin into the mix, antibiotics plus a pain reliever, or an NSAID called indomethacin plus Clostridioides difficile infection, you can see that there is just an overexpression or a much increased expression of all of these different chemokines and cytokines. And they tested this in more specificity in panel B here. In addition, what this leads to is more cells that are recruited to those areas of inflammation. Things such as neutrophils and T cells are increased in the antibiotics plus C. diff plus indomethacin treated animals. And this can also alter the ability of epithelial cells to maintain that tight barrier. So the entire gastrointestinal tract is lined with epithelial cells, and throughout the intestine, it is a single layer of epithelial cells that protect the insides from the outsides. But there are extremely closely associated proteins, a number of different proteins, which form these tight junctions. As you can see in the mock animals here, animals that were not really treated with antibiotics or um, any of the other uh, parts of the experiment. And you see the tight junction pointed at with the arrow here. And in all of the different treatments, those tight junctions remain, except in the animals that are treated with indomethacin, C. diff, and antibiotics. Here, where the red arrows are showing, those tight junctions are starting to basically unzip and allow perhaps some of the bacteria that are going to be in the lumen side of the gastrointestinal tract to perhaps start crossing over that epithelial cell layer. They also saw that there were perturbations in the gut microbiota, that when animals were treated with all three of these different conditions, that the gut microbiome had different constituents. And so this is something that is going to potentially change the way that doctors treat patients who have C. difficile infection. And the senior scientist, David Aronoff, was quoted as saying that ultimately these new results might guide how we treat people with C. diff, particularly with pain management. Right now, it's too early for our results to guide clinical care, but they should be a stimulus for future studies. And this is also shared by members of the research team who are on social media. And this is, I should point out, a really collaborative group of of microbiologists, immunologists, as well as medical doctors who work on gastrointestinal tract health. And so it's really a collaborative effort to make this discovery. All right, moving on. Our next study is one from eLife. And the take home message here is that drug resistance correlates with widespread low level drug use. Now, of course, in order for resistance to occur, there needs to be some sort of antibiotic use that's going to select for those resistant isolates. But what this group wanted to ask is what kind of antibiotic use really drives that resistance to establish itself in a population? Is it low level constant use? And in this case, what they're referring to by low level constant use are perhaps people who might get something like an upper respiratory tract infection or a urinary tract infection who take antibiotics once a year for 5 to 7 days. It's not going to be it's going to be a susceptible strain, so you just take the course of antibiotics, so whatever the doctor prescribes for you. And then the converse of that would be patients who have recurrent infections and who need to get a number of different types of infections treated, or perhaps have repeated infections, such as the C. diff patients that we were talking about in the last story. And so, what the authors hypothesized is that the intense antibiotic use by this latter group, those that have to take many different types of antibiotics or take antibiotics of higher concentrations, those are the patients that are going to drive population level drug resistance throughout a different geographic area. And so they were going to test this hypothesis by comparing the distribution of antibiotic use with antibiotic resistance. And so the first thing they did was to characterize the distribution of outpatient antibiotic use. And here they were able to use claims data from different insurance company, which can distinguish whether many different people are just getting a few prescriptions or whether a few people are getting many prescriptions. And what they found is that antibiotic use is not evenly distributed. This is probably not a huge surprise. But if you look along this line here, along this line, points on this line have three times as many um, for first use or three times as many single use as repeated uses. And so as we start to increase heading up, those are the numbers of people who those indicate people who may have to have repeated courses of drugs. These were then compared to the total antibiotic resistance within different geographical areas. Now, after this, the researchers evaluated whether the annual first use and repeat use are differently associated with population level resistance. And so I'm showing you a multiple regression graph, which they include. This is panel A and panel B are the same pieces of data. Panel B is simply zoomed in. And what they found is that repeated use of drugs actually seems to be negatively associated. You can see that there's a somewhat negative sloped line that you would be able to draw if you were to draw that regressive line, uh, which is negatively associated with resistance when controlling for first use. So they controlled for everyone having a first use, and then they saw repeated use actually has a negative correlation. And so this suggests that it's actually the opposite of what they had hypothesized, that the numbers of people who are just taking antibiotics for a single time frame within the outpatient population may be those that are driving the antibiotic resistance at the population level. So this was highlighted in the Center for Infectious Disease Research and Policy, which is an institution at the University of Minnesota that covers a lot of antibiotic resistance stories. This was also highlighted in a press release from the Harvard School of Public Health, where a number of these researchers were working, and they were able to quote both the senior scientist, and the first author. The senior scientist, Yana Tim Grad, said that our results show that most antibiotic use is occasional, people taking one antibiotic course in a year. And this occasional use is more closely linked with antibiotic resistance than intense repeated use. This is also shared by some of the research team on social media, highlighting some of the takeaways. And what this really indicates is that antibiotic stewardship is an important thing for all people to consider, not simply those who are working within a healthcare system with inpatient patients who are hospital residents and taking multiple courses of antibiotics, but every individual who is given a course of antibiotics throughout the course of a year can affect the resistance establishment in the local area. And so, when you think you have an infectious disease, it's important to make sure that you are giving that infection the correct drug if it needs a drug. All right, moving on. Our next story is from Cell Host and Microbe. And this is a story in which an antibody cocktail is shown that it may protect against all Ebola viruses. Protecting against all Ebola viruses is a big deal because although often in the news it's covered as an Ebola outbreak, there are actually multiple different Ebola virus species, and the treatments for one do not necessarily treat for all. And so, on the left hand side, you can see that all of the Ebola viruses, which are the genus of virus, are filoviruses or filoviridae, of course. But then there are a number of different species of those Ebola viruses. Now, the most common in these outbreaks that we talk about in the news, the large outbreaks, are the Zaire Ebola virus, which is sometimes just called Ebola virus or EBOV. But there are a number of other Ebola viruses which can cause disease. Second most common in outbreaks is Sudan Ebola virus. There's also Bundibugyo Ebola virus. And there are even new Ebola virus like species, which have yet to be confirmed exactly how similar they are. But just a couple of weeks ago, in another study, characterization of a filovirus in China that seems to be related to Ebola virus was published. Now, a number of outbreaks occur based on these, as I just mentioned. And if you look through 2014, this map only covers to 2014, you can see that most of them are that Zaire Ebola virus, the red spots throughout the map here. But that there are, as I mentioned, a number of other species that can cause these outbreaks. The current outbreak is the Zaire Ebola virus, so it would be another red spot, also in the Democratic Republic of the Congo, as is shown here in this country here, this region of Central Africa. And the ongoing outbreak is actually the second largest that's ever happened. Now, it might not seem that large because we only have around 600 cases that have been reported, maybe a couple hundred more at this point. But relative to the West African outbreak of 2014 to 2016, this might seem small because that was tens of thousands of people. But this is still a very large outbreak and one that has been hard to control for a number of months. We've already talked about this in previous Microbial Minutes sessions. And the ability to treat these outbreaks, regardless of what species causes that outbreak, is something that would benefit um, public health workers who are trying to stymie and quench that outbreak as it's occurring. And so that these two studies that were published in Cell Host and Microbe were published at the same time, and they're on the same cocktail. They're talking about two different human antibodies that have already been found to neutralize different Ebola virus species. I believe we've previously covered ADI 15878 and another antibody, ADI 23774. Both of these were um, isolated from survivors of that 2014 to 2016 West African outbreak and were shown to have this multiple reactivity against different Ebola virus glycoproteins. The glycoprotein is the outermost layer of the Ebola virus, and these two antibodies interact with different regions of the glycoprotein, which is important so that they're not competing. Now, um, these antibodies alone, together, before tinkering with them, these antibodies were shown to be protective against Ebola virus in a guinea pig model of infection. Now, the researchers in this first study looked at the FC receptor, which is the constant region of the antibody, and they modified it in a way that improved its ability to recruit natural killer cells and other immune um, uh, functions that will lead to Ebola virus clearance and protection of the patient. And they showed that this modification improved the cocktail protection such that a single low dose of this cocktail was able to protect guinea pigs against two Ebola viruses. They tested against the Ebola virus and the Sudan virus, which caused the majority of those outbreaks, as I mentioned. Now, the second paper looked at this same cocktail, but they looked in two additional animal models to look at the level of protection. They looked in both ferrets and they looked in non human primates. Now, they showed that this modified cocktail was able to protect ferrets and non human primates from three different uh, Ebola virus species. They looked at Ebola virus, Sudan virus, and Bundibugyo virus. And additionally, they showed that it only takes a single dose of a 25 mg per kg injection that would confer this protection, and that's an improvement over current therapies. So you may remember the ZMapp, which is a triple um, antibody cocktail. This is a regimen that requires three different doses of 50 mg per kg, and other antibodies that are currently being tested in clinical trials, such as MAB 114, which requires a single dose but requires 50 mg per kg, or the REGN EB3 requires one dose but 150 mg per kg. And so this allows this lower dose, 25 mg per kg, allows a um, allows the cocktail to go further and protect more people. They also tested different ways of producing the antibody cocktail. They used cell lines, and they also used the plant produced antibody. And they found that either production means produced therapeutically effective antibodies. And so, of course, this is good news. People were excited, those that work both in Ebola basic research as well as those working in public health. And this was highlighted in Science Daily, where the senior author of the second study, the one with two animal models, John Dye, said that this discovery has implications not only for the treatment of Sudan and Bundibugyo virus, but for newly emerging Ebola viruses as well, such as those that I mentioned at the beginning that are still being characterized. This was also highlighted in a commentary that was published in Cell Host and Microbe. We'll link that down below. And what the authors of this commentary point out is that one of the reasons that these studies are able to identify different types of monoclonal antibodies from patients is due to a range of new isolation techniques, which help to find the B cells that are generating the protective antibodies from these patients. Finally, this was shared by the senior author of the first study, Kartik Chandran, who pointed out, I think, an important aspect of these studies, and that's the fact that these antibodies were isolated during that 2014 to 2016 outbreak. And since that time, the structural studies, the neutralization studies, and all of the studies to modify those FC receptors and make these antibodies more effective have been done within 3 years. So that's a really short time from finding a protective molecule to modifying it to showing its protection in a number of different animal models. So really points to the collaborative effort of people who are working within this field. Moving on, we're going to cover a story from Cell, which is the parent company of Cell Host and Microbe. This story has the take home message that quorum sensing molecules can influence phage activity. So this is a big deal because quorum sensing is generally thought to be something that is specific for cells. It's a way that microbial gene regulation can occur based on population density. So, for example, if all cells in a population are producing, let's say, three of these little triangles on the side, three of these quorum sensing molecules, if there's a low population density, this cell is not going to detect the presence of these molecules and it won't be able to change its gene expression pattern appropriately. However, as this cell divides and makes more and more progeny cells, all of these cells will subsequently be producing those three different triangle molecules, and they will be in such close quarters that it increases the probability of a cell receiving one of these molecules and having it interact with different quorum sensing molecule receptors. However, to date, even though studies have shown that this is showing a single species or a single representative microbial type, and there is some crosstalk between different types of microbes. Microbial species can have receptors for other species, quorum sensing molecules. All of these studies have so far been in cells. There have been no studies suggesting that phage can take part in this cell communication means. This is changing now with this study that shows that Vibrio cholerae, which has a phage called VP882, this phage has a gene that codes for an inactivator of a phage repressor that acts in a quorum sensing dependent manner. And so the researchers of this study, there were only two, Justin Silva and Bonnie Bassler, wanted to see how quorum sensing affects bacteriophage activity. And before we get into the exact research that they did, I just want to remind everyone how bacteriophage have different lifestyle choices. After a bacteriophage, shown down here, has injected its genetic material, presumably DNA, this will incorporate into the bacterial cell DNA in a lysogenic form. And then as the bacterial cell divides, it will also replicate the phage DNA and pass on that phage DNA. And this cycle can repeat ad nauseum, creating an entire population of lysogenically infected cells. However, given some sort of stimulus, that phage DNA can excise and start to express the genes that are necessary to generate the different phage particles for assembly, which will incorporate the genetic material and eventually lyse the cell. They'll fill up that bacterial cell so much that they will cause the cell to lyse, and that's why this is called the lytic lifestyle. And so the way that the bacterial cells stay in the lysogenic form is by expression of this CI. The phage has a CI gene. This is going to um, be expressed and make the CI protein, which will then repress the Q gene. The Q gene leads to expression of all these lysis genes downstream. And so by inhibiting Q expression, all of the lysis genes are therefore inhibited as well. Now, what the first observation of this paper is that the cells bacterial cells that were infected, when they were infected with this VP882, and they had the presence of a gene called VQMA, as the cell population increased, there would be lysis of the cells at a given time point or at a given cell population. However, when that VQMA was not present, the cells were able to continue growing, suggesting that this was actually a phage gene that was responding in a population density dependent manner. And what they found was that as VQMA interacts with the quorum sensing molecule called DPO, it interacts and binds and then activates expression of a gene called QTIP, this red gene over here. And what QTIP does is to interact with CI. So here we see the CI protein expression is tagged in red. The QTIP is tagged in green. They overlay. And what's happening is that the QTIP is interacting with CI and blocking its ability to sit down on Q and block its expression, thus activating the lysis genes. This led to a model by which the authors concluded that CI inactivation can occur by two means. One of these has been well documented for decades that when the host DNA is damaged, the SOS response within the bacterial cell can activate REC-A, which will cleave CI. CI will not be present. Q is going to be expressed and will then activate these lysis genes. This second means of CI activation is what the authors in this story have described. This is where VQMA is able to interact with DPO. And when it does, it will sit down and activate expression of QTIP, which then goes on to aggregate CI and prevent it from blocking Q expression, thus again leading to lysis. So, although this is a relatively complex genetic profile, this story was probably the most widely spread of all those that we're talking about because of the implications that a virus can interact with this communication from different bacteria. And so this was covered in Scientific American, for example, where spy viruses eavesdrop on bacteria, then obliterate them. Quoting Bonnie Bassler as saying, as scientists, this is just unimaginable to us. We were delighted and skeptical at the same time. And its first author, Silpa, quoted in NPR saying this was unexpected because it's suggesting there is a virus out there that has joined the bacterial conversation. It was also a number of scientists and science reporters who were very excited about this on social media. And there were all types of very fun analogies, such as spying and eavesdropping, to help people understand how this communication is ongoing. Finally, we're coming to our last story, and this is one from Nature Communications talking about how parts of DNA building blocks can be made in space conditions. So, can life exist in other places? This is the existential question of human existence. Are we alone in the universe, or are there other places that might have similar types of life? And we're pretty sure they have to be similar types of life because of the structure of DNA and RNA, the bond angles that are made, the sizes of the atoms that are included have really prevented any other type of life uh, at the molecular level from being possible as far as we've been able to tell. And to test whether these types of molecules can be found in other conditions, there have been famous recapitulations of some of these organic molecules through conditions that mimic the primordial soup like conditions of early Earth, such as no oxygen and adding a lot of electricity and heat and different types of minerals, reactive compounds. And some of those organic molecules can be generated abiotically, things such as amino acids and other types of organic molecules. Now, the question is about sugars, specifically because sugars are part of the backbone of nucleic acids, which make up the code of life, right? So, if we're looking at RNA, then we're talking about ribonucleic acid. And if we're looking at DNA, we're talking about deoxyribonucleic acid. The difference being simply whether there is this hydroxyl group on the 2' carbon here. You can see that it's present in RNA, but not in DNA. And ribose has been identified in experiments simulating the conditions of space. So, photoprocessing of ices, meaning that there would be a lot of UV exposure in space. Of course, there's no ozone layer to prevent UV exposure. It's going to be extremely cold, and these conditions have been used to generate ribose um, from precursors. Now the question is, what about 2-deoxyribose? And that question is present for a couple of reasons. First, being that DNA is inherently a more stable molecule than RNA, and so the researchers took sugar alcohol. They took isopropanol and added it with water at certain concentrations, which they then incubated at very cold conditions. And they also exposed this to UV light and then tested by mass spectrometry to see whether or not deoxyribose had been generated. And so here is the standard. This is what the peak pattern of 2-deoxyribose looks like. And you can see that in their sample, they were able to recapitulate all of these same peaks. And they can increase the size of these peaks by using a heavier isotope, using C13 instead of C12, which is the normal isotope for carbon. But by simply increasing the weight slightly to C13, they are able to shift all of those peaks, showing their specificity for carbon, and that these are indeed generated um, through the process by which they had generated the 2-deoxyribose. However, when they tested meteorites themselves, they failed to find any 2-deoxyribose, which they say might be because of the very low concentration or because of the relative instability of the products which they were able to find. Now, the idea of having DNA precursors in space is quite exciting. This was also covered by a number of different outlets. It was covered by Cosmos. It was covered by the UK-based newspaper, The Independent. And this is really a story of two different science communication means. So in Cosmos, the title or the headline is that DNA sugar can form in space, experiments show. And The Independent uses the headline that proof DNA building blocks exist in space points to key role for meteorites in creation of life on Earth, which is perhaps not using the type of language that scientists themselves would like to use, since there are no DNA molecules in space. In fact, when the scientists tested the meteorites, they did not find the 2-deoxyribose. What they did show is what the headline in Cosmos says, that the DNA sugar can form in space, not that it does form in space. And I know that's just the difference of one verb, but that difference is very important in interpreting what the results are. So the authors wrote that the presence of sugar derivatives in primitive meteorites, together with other compounds of biological interest, such as amino acids, nucleobases, and amphiphils, is consistent with a scenario in which a significant fraction of the inventory of compounds from which biological processes started on primitive Earth may have been delivered from comets, meteorites, and interplanetary dust particles. So perhaps the source of our DNA and our organic molecules may have come from space, according to what the authors are writing. And the Independent did walk back their headline. They wrote specifically, though the scientists could not definitively confirm the presence of 2-deoxyribose on the actual meteorite samples tested. So anyone who goes on to read beyond the headline would have gotten the full story. But of course, we live in a headline centric world, so it's important to get every part of the news article accurate. All right, that's going to wrap it for this show. We've covered 5 different important stories. One is that common painkillers may exacerbate C. difficile infection. We also learned that drug resistance correlates with widespread low-level drug use. We talked about how an antibody cocktail may protect against all Ebola viruses, which is very hopeful and positive and optimistic given the current outbreak. We discussed a quorum sensing molecule and its influence on phage activity. And finally, we talked about how parts of DNA building blocks can be made in space conditions. It's going to do it for today's Microbial Minutes. Be sure to subscribe so you never miss another show. We'll actually be back next week after simply a week's hiatus with our next Microbial Minutes.